I think most people are broadly familiar with the general life cycle of a mushroom. It starts with a mushroom like this. These are shiitake mushrooms growing out of a substrate log. And everyone knows, right, that the mushroom is a structure that carries spores. And the gills on the underside of the mushrooms are the structures that shed spores. Spores are very tiny, almost microscopic gametes, basically. They recombine sexually to form a mycelium, the vegetative part of the fungus. And this is the white mycelium. It consists of very tiny thread-like strands. And this I got from the bottom of a log today. But typically in shiitake production on a log, we inoculate with these mycelial strands, not with spores. Spores are not involved at all. These mycelial strands colonize the log, and eventually they give rise to the mushrooms from the log. So if we're trying to if we want to understand the process of shiitake mushroom production from start to finish, basically it starts with developing a mycelial culture. And here, for example, we can take one of these shiitake mushrooms, and we don't want the spores. We want the sterile tissue inside the mushroom, and we just split it apart. And by the way, we're working in a sterile air cabinet, not just out in the open like I'm at right now. So we have that tissue inside, and it is completely sterile. We just take a small amount of it with a sterile scalpel, and we place it on a nutrient auger. This is called potato dextrose auger. It allows the fungus to grow. There you can see just a small portion of the interior of the mushroom. We seal this off and incubate it for a few days, and it will begin to grow mycelium, just like you saw in this little fragment here. Here's a plate that's been growing for some time. It's covered with the white mycelium. These little chunks taken out were for transfer purposes. And once we have the culture, like at this stage, then we're ready to transfer it to a secondary substrate, and that would be this. This is grain, sterile grain, and once again, working under sterile conditions, we would take this culture that is covered with mycelium, cut it up into small pieces, and transfer it to this sterile grain. And then we'd seal it off and shake it around to mix up that mycelium in the auger with the grain, and allow this to incubate for several weeks. It will be covered with the white mycelium once again. And then the last stage, the stage at which you would be most likely to acquire it, would be after it's transferred to sterile sawdust. This is just hardwood sawdust. This is a bag of hardwood sawdust to which the grain inoculum has been added, shaken around, and it has been incubated for several weeks. And you can see the white mycelium just covers the entire bag. And this is what you then take a small portion and put it into the hole that you've drilled into the log. And eventually, as I say, that mycelium colonizes the log, and a year later gives rise to those mushrooms.